Uh, I know that the main audience are from in Thailand, and they are also international, and then the variations in the languages and accents. Uh, Anytime you feel something, I'm a speed, and then not understanding, kindly make a note, or you can also raise a, just give some kind of a signal. I will repeat it again. So yeah, we can move forward. And then the intention of this talk is to make sure that everybody understand the values of scientific developments in medical cannabis, its impact on the society as well, because several countries has already legalized, but there are countries like Thailand and then Southeast Asian countries are legalizing. Um, we need to know the gaps that varies importance priorities that we need to give, and then it's not just a jump into the conclusion. Oh, everybody should get the licenses, but after getting the licenses, what is the importance? How you want to jump into the biopharmaceutical industry? It is not just a recreational. I'm not here talking much into the recreational aspects, but I'm more into the scientific aspects. My name is Srinivasa Reddy. I'm a CEO of Phytotech and also CEO, the founder of two companies, which is Phytotech Innovations, which is more into the technologies of the control environment. I'll explain to you what is the role later on stage with the case studies as well. Uh, the innovation in a control environment means the controlling the plants in a domestication of a plant from the wild varieties, or bringing the compounds into the higher level of efficacy. That's the role of the phytotech innovations. Extract the biologicals is a post-harvesting technology, which means that you extract the compounds, which is available 560 compounds in the uh, 560 compounds, and then how we can extract the specific compounds and use the conjugate molecules for the medical formulations or innovative the compounds, conjugate molecules for the different drug delivery systems. So that's the role of extract biologicals. You can, if you can see symbols. And the Leaf Cross Biomedicals is a company where they grow the controlled environmental cannabis, one of the premium qualities in the Kootenay areas of Canada, which is called Nelson. And they also do the processing and formulations, which is a scientific technologies has been led by me. And then I'm acting as a COO and CSO together. So these are the three companies that I am involved completely and then working on. And the establishment of the phytotech and extract biologicals in Ontario, Toronto, and then Leaf Cross Biomedicals is in British Columbia. Going to the next, yeah, this is just a little bit about my background working in the two PhDs related to the plant science and the technology. One of them is in the plant nutrient metabolic engineering. The other one is related to the controlled environmental technology. The one of the masters is in cancer research, where the plant-based molecules can be used for the cancer curing, which almost related to the Ayurveda and then ethnopharmacology. And then other masters is on uh, please expand. Okay, okay, yeah, okay, sure. Just give me a second. Sorry for that. Not giving an option for the expanding the full screen. Okay, great, great. Hope this is okay. Okay. Uh, yeah. So that's the background, and then one of the PhD was from the South Korea, and then all other related in the Canada, and then India. So with a huge the scientific background and also the plant-based informations and then technologies that whatever we have, we wanted to contribute into the research and development side, more into the biopharmaceutical pre-harvesting and the post-harvesting technologies in the cannabis industry. So which I worked almost like a 6 years now, doing the consultations all over the world as well. Um, so coming into this is the things that I explained at the beginning itself, which I don't want to waste your time. So my role is coming as a scientific guy, coming as a technology guy that can bring the values from the connection, the bridging between the conventional the knowledge of the cannabis, because the cannabis is existing for the millions, the hundreds of years, which is used also unknowingly or unknowingly used for the different purposes by the humans and the different animals as well, including the microbials. They always have the evidence that when the microbes go and sit on the cannabis plants, they can reproduce and then they can survive for the more than 40% of their lifespan, healthy lifespan, which you can understand what is the value of the every compounds existing in the plants, which we don't know that we are expanding right now. But our role, my role as a scientific guy, to come into the situation right now, connecting a bridge between the conventional knowledge to the futuristic biopharmaceutical scientific knowledge of the plants and its compounds to the potential use for the health benefits. We also have the adverse benefits from the unknown dosage systems or unknown the cultivars, the strains you take it for the different diseases or the different issues. That has an adverse impact. So here, the tailoring specific size of the receptors and the specific molecules is more important. That's how the connection between the scientific guys and then MDs and then doctors and those are the relations. We wanted to make sure the molecules are setting it to the properly to the targeted diseases, and then they can prescribe it. That's the main role of the conventional knowledge connecting to the scientific knowledge. So going to the second stage, the so far the cannabis is a highly exploited plant. I use this word all the times in my talks. Highly exploited, least explored, which means that 26,756 publications has been done so far, which is comparatively to the tobacco more than 1.8 million publications so far. Tobacco has one nicotinamide, and then tobacco has some other compounds. Basically, they're the smoking things, and then it has the adverse impacts, but still people taking advantage of publishing so much, and the governments are granting so much. But now there is a problematics in giving the government fundings for the cannabis research, even though it has a 560 compounds. It usually until the last couple of months, it's a 480 compounds. Now it's confirmed that 560 compounds because there is a research publications as well, and those compounds are difficult to identify right now as a names, but it's already existing. It's called macrocannabinoids, microcannabinoids, and the nanocannabinoids. The supportive system of macrocannabinoids is very much dependent on the nanocannabinoids and the microcannabinoids. I'll explain you further this slide. So when you talk about what are the areas of research has been done in cannabis till yesterday, that's why my slides were a little bit delayed, just to get the accurate values of publication.
That's a great question.
Which is from the source is from Web of Sciences, which is a highly valuable scientific the publications bank. I can say the knowledge of the things. So the psychiatric issues is highly published and the highly researched. And the least researched, if you go to here, the plant science, only 704. So how many people talk about the plant science, internal science of the metabolic pathways, and then you talk about the impact of the pathways of every coding 560 60 compounds, how it is coded, how it is targeted to enhance. People come with a just they're making a genetic the GMOs or changing the plant's properties by just propagation, cutting the plants and then making. So it is not the way that we wanted to work on. So the way is understand the pathways of each and every compounds, and then go and target the compounds, enhance the compound. It's not the THC, it is not the CBD. It's beyond those kind of compounds. That's why we wanted to focus majorly on plant science as well, medicinal and the legal. So the psychiatric research is an adverse impact and then how it affects, but we need to enhance the compounds to cure different issues here. So that's the chart, the pharmacology, pharmacies around 3034. We need to focus the areas. We need to really give the benefits to the human being right now with the scientific evidence, and then the social benefits. It completely depend on the giving the fundings and the giving the research and the priorities on the plant science, biochemistry, and then different pathways, healthcare sciences, and then those kind of things. And then even environmental sciences is a highly important, which is 274 publications till date. Is extremely right where I'm placing. Because the environmental, the science will impact on the plant's growing conditions, which means that when you domesticate the plants, it means 5% of the variation in the controlled environment will give you the 5% variations in the final products in the plants. Oh, so when you talk about 5% of variations, which can trigger the inhibition of majorly inhibition of so inhibition of the majorly significant compounds. So that's the chart that it indicates. And then let's go to the other slide. These are the some of the compounds that how it has chances in the metabolic pathways, but coding of the metabolic pathways. Every 5% of the variations can give the changes one of the molecules. Here I mentioned about whether it's a hexonic acid or the hexanoyl acid or the oleotolic acid. So these are all very important parameters to produce the CBDA, THCA, CBDC. So if you're missing, for example, melanyl, it's a coenzyme, like enzymes that triggers the factors of the CBDA, CBDC, whatever that goes and then impacts on the production of the final compounds, which is where we are targeting about the cannabis and then the quality, consistency. So going to the next slide, I'm talking about once we use this type of formulations and then in the plant production, the terpene biosynthesis or cannabinoids biosynthesis, we need to understand and we need to go, once we extract the compounds, we have a different cannabinoids demonstration. When you have a domesticated plant, you can target on the terpene production, entourage terpene effects, because when you have a plant, and then it is not just one type of a terpenes. It has a multiple terpenes. When you combine the terpenes itself, it has a different type of entourage effect. And then you're talking about the inter-entourage effect, which is the phytocannabinoids and the terpenoids, because the plant does has the terpenes, cannabinoids, and phytocannabinoids. There are the terpenes and cannabinoids and the different types of cannabinoids inside the plant itself. That's where we need to work on to give the priorities on how we can understand and then address the compounds in the plant, in the human, to the diseases. There is another clinical study towards the herbal medications that I mentioned. This is a recent studies in 2020 October, I think early October, end of the October, which suggests around 560 compounds in the plant, and then out of the 560 compounds, 120 cannabinoids, which is not just a THC and the CBD, but it is beyond the THC and the CBD. So we have a huge, um, huge benefits understanding the plant's scientific evidences. And going into the other slide, oh, okay, that's the last one. Okay, so yeah, the cannabis pharmaceuticals is entirely depending on the pre-harvest and the post-harvesting technologies. Just to give you a conclusion, and then evidence-based studies where you can talk about each and every strain is addressing the different types of medical issues. So anybody can come and then say, okay, take the other strains, and then without the knowledge, it is not accurate. I think it takes like a 2 to 3 more years to know the compounds available in the plants and how to address those compounds to the human or the veterinary issues, and then kindly, um, you know, how it yeah, we need to give the priorities and understanding the scientific aspects. That's why we need to value whoever is in the scientific field rather than coming from the I have a 20 years of experience or a 12 years of experience in a street growing, and they can come and suggest, because there is a one study from the Harvard University, probably Dr. Sandra Carillo, she knows because we worked on a couple of things, and then the sperm count of a human is depend on what type of strains are they taking about taking into the smoking or the ingestion. So one strain inhibited the sperm count, and the other strain enhanced the sperm count. So it is just like a any other sleeping pills or any other medications. It does has a negative impact as well if you don't understand the plant, if you don't understand its impact and the purpose you're taking about. So that's why I think it's a great platform that CISW has given me to talk about a few minutes. And future, I'm definitely able to collaborate with you.
So is it here?
Yeah.
You see the audio?
Perfect.
So, yeah.
So you see me there now, right? Okay, thank you, Mia.
Can you hear them?
So it's good. Thank you. Thank you, everyone.
Okay, great. Thank you so much, Mr. Reddy. I just wanted to touch on one of your main points. Actually answered the question I was originally going to ask. I was going to ask what are some of the significant discoveries that you've made while isolating these compounds that can be applied in society? And then you mentioned that isolating certain strains can impact sperm in human subjects. So that is one example. Can you give us another example of these different uses that can be introduced in society by isolating these different compounds with such results?
Yes, there are compounds. I mentioned you 3 types of things. Number one, macrocannabinoids and microcannabinoids, nanocannabinoids. Okay, so the macrocannabinoids is just like a bricks. Microcannabinoids is just like a sand, and the nanocannabinoids is just like a cement. Just when you're constructing the wall. When you're talking about THC, it's decarboxylated THC that go directly into the central nervous systems, which is unavoidable. Immediately it goes. But what about you have some? I mean, not you, but somebody, the subject X has an issue on the kidney or on the chronic disorders in the lung cancer or something. So the molecules has to go target on the receptors located in the kidney or the lungs, right? So that's why it is very important. It's already proven 1 or 2 studies. Probably if you can take my the email address here, I know that we have a very least time to talk about, but you can contact me directly, and then we can work on. I can answer everything. I can share. Maybe I have a screenshots that in front of me, nearly 1,500 different types of researches, and out of that is 10% of them are related to the cannabis impact on the human being, negative and the positive both. I can share with you a couple of research papers. Yes, it has on the stress factor as well. A different level, different compounds has a percentage of compounds you take it it has a bioavailability. That is proven as well from our collaboration research. And our collaboration research also worked on a cancer research, how bioavailability of the molecule, what are the sizes in the different plant strains. Because it's a THC, end of the day, everything is a THC, but the THC also has a different sizes of the molecular sizes. So you don't need to nanotize the molecules. When you extract it, you don't need to nanotize because you already have a naturally made a bioavailability comparing to the other different strains. So this is another example.
That's fantastic. Very interesting information that you shared with us.
Yeah. That's why understanding the plant is absolutely important, because bioavailability, when you do it a manual, then like as a synthetic bioavailability using the sonication or some other technologies, nanotization, there is a probability that your molecules will break. And then as I shown here in the slide here, if you can see, the molecules of the enzymes can break, or the structural aspect of decarboxylation in the THC is this portion is decapsulated. When they decapsulated, it activates as a higher, it triggers into the central nervous system. So if we can work on non, damaging the molecules with a understanding the plants itself directly, and then what is the molecular sizes, then definitely we can go and then find out natural cure to any type of diseases. This is our intention of the research and development right now, which is not done by a lot of people.
Okay, thank you so much. And years of studying and research into this, we've got to move on to our next guest. Thank you so much, Mr. Reddy.
Thank you so much. Thank you. Appreciate it. Yeah. Bye-bye.